Good evening. My name is Corrado Paina, Executive Director at the Italian Chamber of Commerce of Ontario, Canada, ICCO Canada. Welcome, and thank you for joining us today for the third webinar organized by ICCO Canada in partnership with AIRI, International Association of Italian Researchers, and the Sistema Italia, Embassy of Italy to Canada, Consulate General of Italy in Toronto, Italian Trade Agency, Istituto Italiano di Cultura, and Italian National Tourist Board. Tonight, we'll talk about a particular group of diseases that affect at least 10% of all adults, the genetic disorders. As the scientists unlock the secrets of Canada, they're learning that nearly all diseases have a genetic component. Some diseases are caused by mutation inherited from the parents and are present in an individual at birth. Other diseases are caused by acquired mutation in genes due to some environmental exposure. These, for example, include many cancers. Scientific advances in the field of genetics and the study of DNA today have obtained a lot of information of great importance for our health, such as any genetic predisposition or the importance of environmental factors. Before introducing today's speakers, I want to take a moment to thank Sistema Italia for their strong support to ICCO Canada initiatives. We have a message from the Consul General of Italy in Toronto, Eugenio Sgro. Thank you.
Good evening. As Consul General of Italy in Toronto, I'm glad to introduce the webinar devoted to genetics and hereditary disorders organized by the Consulate General of Italy in Toronto in collaboration with the Italian Chamber of Commerce of Ontario, the Embassy of Italy, the Italian Cultural Institute, the Italian Trade Agency, and ICCO, an association of Italian researchers which forms a networking platform for thousands of Italians that study and hold research-related jobs from around the world. Together today, we will deliver into the complexity of genetic diseases and learn more about recent progress in this field. It's widely known that there are thousands of genetic diseases. Currently, over 6,000 have been classified, and new ones are constantly being identified in medical literature. In fact, estimates indicate that genetic defects are present in at least 10% of the world's adult population. Additionally, approximately 1 in 21 people have a genetic diseases considered rare. In Italy, there are about 2 million people affected by rare genetic diseases, 70% of which are children between the ages of 0 and 5. Unfortunately, about 30% of rare diseases patients do not have a diagnosis and are likely to live with a condition that will remain unidentified. In many cases, in fact, we lack the interpretive key necessary to recognize the origin of the symptoms affecting these patients. The role of research is therefore fundamental in gaining a better understanding of the origins of these pathologies, as well as in ensuring effective diagnosis and in developing adequate treatments for the millions of patients affected. It's precisely for these reasons that an event like the one we're attending today, which is part of the ICCO AIRI lectures, is extremely important, and even more so today, given the complex times we are currently experiencing. Italian research institutes have proudly made important contributions in the field of genetic research, and Italian universities have trained thousands of researchers now active in the international arena. Among them, surely, stands out Dr. Stella Lanni, whom we are honored to host as our speaker today. Dr. Lani holds a PhD in molecular genetics from the University La Cattolica in Rome. In 2014, she moved to Toronto to study the molecular mechanisms involved in a particular group of genetic disorders responsible for many neurodegenerative and neuromuscular diseases. Dr. Lani, a researcher at the Hospital for Sick Children in Toronto, represents an example of Italian excellence and a reason to have great hope in the future science. Thank you for your attention. Enjoy the lecture.
Consul General Gro, thank you very much. I leave the floor to Dr. Emilia Luca, PhD Research Associate at Sunnybrook Research Institute, Toronto, for a brief introduction of AIRI.
Good evening, everyone. I'm Emilia Luca, co-founder and main referent of AIRI Toronto chapter. Before presenting our association, we would like to thank the ICCO Canada for organizing these scientific lectures and today's sponsors, in particular the Consulate General of Italy in Toronto. AIRI is the International Association of Italian Researchers. It is a not-for-profit association and is one of the largest scientific Italian communities in the world. We are more than 25,000 researcher members, and we have 8 chapters in 7 different countries. Our mission is to promote scientific collaborations, exchange of professional information, and cultivate the role of scientists in popular science. We know science, we do science, and we want to communicate it clearly and effectively to the public. If you would like to know more about AIRI, follow us on social media. Before leaving the floor to our speaker, AIRI Toronto chapter congratulate a honorary member of AIRI, Professor Giorgio Parisi, that yesterday received the Nobel Prize in Physics for the discovery of the interplay of disorder and fluctuation in physical systems from atomic to planetary scales. Now I pass the floor to Dr. Stella Lanni, who will present and explain us the important role that genetics play in our lives. Thank you for your attention and enjoy today's talk.
Hi everyone. I'm Stella Lanni. Thank you, Emilia, for giving me the floor, and thank you for all the nice introduction. And everyone, thank you for being here tonight. So let's start with what is genetics. Genetics is the study of how traits, or to better say genes, are passed down from one generation to the next. Our genes carry information that affect our physical characteristics, but also our health and in some ways also our personality. Each humans start off as one single cell and grow into trillions of cells. The information necessary for the cells to become a full embryo are all inside one molecule, the DNA. DNA contains all the information cells need to make you from head to toes. What is DNA? DNA is made of chemical blocks that are called nucleotides. They bind to each other and they stack on top of each other, forming these double-stranded helix. To simplify, let's think of DNA as a book that's written using an alphabet that's made only of 4 letters, A, T, G, C. The combination and the different words we can make with these 4 letters are many, many. And pieces of these letters, so pieces of DNA, will make what we call genes that we can consider as sentences in a book. And you should think that when we estimate that in human cells, there are around 25,000 to 30,000 different genes. So genes contain the instructions the cell needs to make proteins that are actually the workers. So proteins are the molecules that will actually do all the function that each cell has to put together. So neurons, eye cells, skin, gut cells, and whatsoever. So basically we start with DNA, with what's written in our genes. As the cells is not able to read what's in the DNA as it is, we need to go through a translation forming a molecule that everyone heard about lately, which is the RNA, that has the same molecular blocks similar to DNA, but it is single-stranded. From the RNA, the cell is then able to make proteins. And so the workers that will allow us to be what we are and will allow our body to function. So the DNA is stored within the cell nucleus, and the cell nucleus is approximately 6 micrometer in diameter, so very, very tiny. And you need to think that in each cell, the whole molecule of DNA is about 2 meters long. So to make the whole molecule of DNA able to fit within such a small place, the DNA is rounded about specific proteins and is compacted in these structures that we call chromosomes. Most cells in our body have 2 copies of each chromosomes. So we will have 2 copies, one derived from our mother, one derived from our father. These cells are called somatic cells. And so in each of those cells, we have 46 chromosomes forming 23 pairs. But situation is different for sex cells. Eggs and sperm, in fact, they only have 1 copy of each chromosome. Why? Because when an egg with one set of 23 chromosomes is fertilized by a sperm cell with another set of those 23 chromosomes, we will have the formation of a diploid cell, so a cell containing the full set of genes that are necessary for that fertilized egg to become a full embryo. Here I'm showing you the chromosomes, how we historically looked at them. They're very cute in my opinion. And here you have a female karyotype with the 22 set of chromosome plus the sex chromosome. Male and female karyotype are absolutely identical, but for the sex chromosome. So in female, we have 2 chromosome X, while in male, we have 1 chromosome X and 1 chromosome Y here. And all the information, all the genes that are stored in this little chromosome Y are always necessary for an individual to be a male. So how is sex inherited? We said that sex cells only have 1 set of chromosomes. So all the eggs in a female will have 1 chromosome X, one or the other. In a male instead, sperm cell will have a chromosome X or a chromosome Y. So progeny will have a 50% chance to be born as a male or as a female. And this is true for every other chromosome. So if we consider this little schematic as 1 chromosome, let's say chromosome 1, in which, for example, we have this orange gene. So we said gene are sentences. So let's imagine this gene says, the orange gene says, the cat ate the fat rat. This female has 2 chromosomes with the same orange gene. In this male instead, some sperm cell will carry the chromosome with the orange gene, but in other cells, this sequence of DNA mutated. So now the sentence will read as, the cat ate the fat rat. So the progeny will have a 50% chance to actually inherit the orange or the blue gene. So to inherit the mutated or the mutated gene. And if we think of the blue gene as a gene that might contain a mutation that's actually pathogenic and will lead to disease, here we have the basic or how diseases are actually transmitted to progeny. Genetic disorders are in fact diseases that are caused by changes in the DNA sequence. They are hereditary only when they occur in a germ cell. So if we have a mutation in a skin cell, for example, it will still lead to a genetic disorder, but it will not be inherited. When instead we have a mutation in the germ cells, so in eggs or in sperm, here now we have that disease to be passed on further generation. In genetic disorders, we can have mutations occurring in 1 gene or in multiple genes. Genetic disorders are rare, but there are thousands of those, and they represent 80% of all the rare diseases. So the most common health problems, for example, heart disease or diabetes, obesity, hypertension, autoimmune disease, cancer, or atherosclerosis, they do not have a single genetic cause. They're called polygenic disorders. So multiple genes are responsible for those diseases. And a great role is also the role of lifestyle and environmental factor. If we want to do an example, let's think of a person who has been a smoker for the past 20 years of his life. This person lives in the same neighborhood of another person, and they go around together, so they have a very similar lifestyle. They eat almost always the same kind of food and whatsoever. Person 1 will actually develop a lung cancer, person B will not. Why is that? That's because person A inherited a set of other genes that probably have mutation that together with the mutation that are induced by smoke will actually induce and give lung cancer. Person B instead is protected by its genetic. But the truth is we don't know everything that's involved in polygenic disorders. They do not have a clear pattern of inheritance, and we don't have all the genes identified. So what I'm trying to say here is lifestyle, it's really truly important together with your genetic. Polygenic disorders often cluster within families, but the incidence in relatives of people with such diseases is estimated to be around 2 to 4%. While the incidence in relatives of people affected with monogenic disorders that I'm going to give you an example in the next slide, is about 25 to 50%. So let's look at one monogenic disorder. So disorder that's due to mutation in one gene. For example, cystic fibrosis. Cystic fibrosis is the most common life-threatening inherited disease in children, and has an incidence of 1 to 2,500 kids, 1 in 3,600 in Canada. It's a multisystemic disease, but most of the symptoms actually involve the lungs, where we don't have the clearance of this mucus as it happens in healthy individuals. And the formation of this mucus in the respiratory tract gives rise to congestion and bacterial infections, and often to pneumonia. And also we have very severe symptoms affecting the digestive system, like malabsorption of nutrients by the pancreas. Cystic fibrosis is caused by mutations in one gene that's called CFTR gene. And it is an autosomal recessive disease, which means that a person needs 2 mutated genes to actually show symptoms. So here we have a family where both parents are actually carrier of a mutation in the CFTR gene. They are not affected by cystic fibrosis, but 1 out of 4 of their kids will actually have a chance to inherit both mutated allele and actually have the disease. So if, for simplicity, I put here the same schematic that we have seen before. So we will consider capital C as the non-mutated allele and small c as the mutated one. So you will see that 1/4 of progeny, so 25%, will have normal genes and will not be even carrier of the disease, while 50% will be carrier of the disease, and 25% of progeny will actually have cystic fibrosis. So cystic fibrosis, as I told you, is an example of autosomal recessive disease, but we have another kind of family of diseases that are called autosomal dominant diseases. What it means is that you need only 1 copy of the mutated gene to actually have the disease. And amongst this group of diseases, we have repeat expanded diseases that I'm going to be talking about a bit more because it's my area of expertise, and it's they are the diseases we study in Dr. Pearson lab. These diseases are caused by mutation in repeated DNA. So if we go back to the example of gene as a sentence, the cat ate the fat cat rat, I showed you what's the standard kind of mutation, where we have only 1 letter that will actually change. What happens in a repeat expanded disease is very different. Here, we will actually have an increase of the size of the repeat word. So now we will have the cat ate the fat rat. And in the next generation, this word will actually be expanded even more. This is a big problem because this expansion will be also present in the RNA, and the protein that will be formed will actually also have the expansion, and will be a protein that's not able to actually do properly its job and will give rise to disease. So in repeat expanded diseases, we have the transmission through generations, and we have an increase in repeat size going from one generation to the next. Also, we have a reduction in the age of onset of the disease. So let's say in the first generation, symptoms will start around the age of 70, 80. In the next generation, we start around the age of 50, 60, until we have juvenile or a childhood onset of the disease. And in some kind of diseases, we also have a congenital form with symptoms that show already in utero. Also, we have an increase in disease severity. So if in the first generation we have very mild symptoms that show late in life, while we go through the generation, we will have very, very severe symptoms, and often that in the 2 first months of life. Repeat expanded diseases are many of them, and they are responsible for almost 50 human diseases. One of the most frequent is actually Huntington's disease. Huntington is a neurodegenerative disease affecting 3 to 7 every 100,000 people of European ancestry. In different populations, the estimate is a bit lower, and 1 to 7,000 people in Canada. So here we see an image of an unaffected brain, and here we see what happens in people affected by Huntington disease. We actually have death of neurons. And that is correlated with symptoms that start off with a change in personality, depression, and will go with inability to actually being able to speak or to actually do proper movements. Also, another characteristic symptoms of Huntington disease are these movements of the hands that are called choreic hand movements. Diagnosis usually happens around 30 to 40 years of age, but we also have juvenile onset of Huntington disease, and that occurs 15 to 20 years after diagnosis. There is currently no cure or treatment existing. The cause of this disease, as I told you, is an expansion. So in unaffected individual, we have a CAG tract that's repeated up to 36 times within the gene that's called Huntington gene. In affected people, this repeat goes further, way more than 36 repeat, and it gives rise to an expanded RNA and a protein that's actually mutated and will lead to disease. So in the Pearson lab at the Sick Kids Hospital, they identified some very particular structures that I'm showing you with this schematic that are present only in this expanded DNA. And you can see here images where I show this is a DNA fragment, and here you can see these structures that are present in different tissues of patients affected by the disease. So as these structures happen only in the puntated allele, we thought of using them as a target to try to modulate the expansion. So here we have our expanded DNA with the structure, and this is what usually happens, right? So the expansion will get bigger and bigger and bigger. What we want to do is actually stop or reverse the expansion to try to stop or reverse the symptoms of the disease. And we recently published a paper where, using a small molecule, a compound that's called NA, that binds very specifically these structures, we are actually able to accomplish that. So we are actually able to let the cell process this structure to lead to a contracted, so a smaller tract instead of a bigger expansion. These results were very promising. Also, we had very nice results in vivo in mouse models of Huntington disease, and we are now moving forward hoping to see if this molecule can really help patients one day. Another disease we study in the lab is amyotrophic lateral sclerosis, which is a progressive neurodegenerative disorders that affect nerve cells and causes loss of muscle that are necessary to move, speak, eat, and breathe. What I'm showing you here, all of these dots you see, color dot, they're all genes that were identified to be mutated in people with amyotrophic lateral sclerosis. The bigger is the dot, it means that more people present that kind of mutation. So as you see, there is really a variety of different mutations that are causing ALS. One of the most frequent, so here the big yellow dot, is the C9orf72 gene. And the mutation that causes ALS in this gene is actually an expansion. So we have a repeated tract that is expanded hundreds and hundreds of times in this individual and actually gives rise to the disease. So in the lab, we observed that this expansion at the C9orf72 actually gives rise to the same kind of structure I showed you before. And as this is a very complicated sequence to look at, like technically, we have now up and running a technique to be able to actually measure the size of this repeat. So here, what I'm showing you, this band here is the non-mutated. This is cell lines from a non-mutated person. This is the DNA of that region, and you can see the size is very low. Here we have a patient in which you do see that there is one gene that has the non-mutated copy of the gene, while the other one that's carried in the other chromosome has a bigger size. So it actually has the expansion. So now that we know that we have in ALS the same kind of structures we observed in our other diseases, and now that we know that we are actually able to look at those because it was not easy on a technical point of view, now we are trying to use a similar approach we used with Huntington, so use drug-like compounds to try to modulate the repeat track. But I'm not showing you the results as they are still in progress. Lastly, I want to talk about myotonic dystrophy type 1. This is a progressive muscle degeneration that gives rise to muscle weakness, inability to relax muscle, difficulty breathing. It's caused by an expansion, and this time we have a CTG sequence that's expanded within this gene that's called DMPK. The prevalence of DM1 worldwide is 2 to 14 every 100,000 people. But it's 20-fold greater in Saguenay region in Quebec. So in a sense, it's a very Canadian disease. And I just wanted to point out that we have collected these very rare cell lines from these individuals that are members of DM1 family. So they do have myotonic dystrophy in their family. But instead of growing bigger and bigger with generation, as I told you up to now, what happens is this family is that the repeat size actually gets smaller and smaller with generation. And as a matter of the fact, the late generation, like the great-grandson, actually has symptoms very late in life. So we don't know why that happens, but obviously these patients are very precious on a research point of view, because if we identify what other part of the genome is contributing to this contraction, we can try to use that same mutation or that same variation in the DNA to actually modulate repeat in the expansion families. And another thing we are working on is the congenital myotonic dystrophy. So as I told you, there are cases in which we have symptoms of the disease already very early in life. And in the congenital myotonic dystrophy, actually these babies show symptoms of the disease already in utero. They often die between the first few months of life, but when they survive, they have very severe symptoms, and the muscle symptoms go together with intellectual disability usually. So we were lucky to work with clinicians that were able to collect samples from 19 families. And we had 20 samples from people with the congenital form of myotonic dystrophy. I'm not going to show you the whole table, but I just want to point out that we studied this modification of DNA that's called methylation. Is a chemical modification of the DNA, and you can see that it's the value it's almost zero for all the controls and the classical DM1, but for the congenital patient. Why it's that important and what's the role of the methylation in the congenital disease, we don't know yet. We are working on it. But what's important is that we are hoping to be able to use the presence of this methylation as a tool to actually being able to do a prenatal diagnosis, because currently there is no way for the families to know if their kid is going to be affected by classical myotonic dystrophy or by the congenital form, which is way more severe of myotonic dystrophy. To do that, we need to collect samples from more families, obviously, and that's why the role and the participation of families together with clinicians and researchers is so crucial for basic research. So to finish, I just wanted to show you and to present to you this tool that I think is really important. It's the orphanet website. It's the portal for rare diseases and orphan drugs. Here you can find information regarding all the rare diseases. And you can find information regarding orphan drugs, so drugs for very rare diseases, or you can try information regarding the clinical trials that are ongoing. You can check what kind of research is being moving forward for a particular disease. And what I think is also really important, at least based on my experience when we meet families of patients, is the directory of patient organization. So here you can meet other people, other families that are going through the same problems, and I think it really makes the difference. Thank you, everyone, for listening. Thank you to the organizers, and then I'm here to take your questions. Thank you.
Okay, Stella, very interesting talk. We have some questions coming. I give a preference as question, but there are a lot. As we receive one copy of genes from our mother and one from our father, shouldn't we all look more similar to our siblings? What makes us all so different?
Yeah, so this is very interesting question. Thank you. So what makes us different is that it's not all that simple, as I've showed you. So what actually happens is that while the two chromosomes, one from the mom and one from the dad, get together, there is actually an exchange of informations between the two. That's called crossover. So that allows us for genetic variations and allow us to actually be all very different from each other.
Another question is, why are symptoms so different from one person to another for repeat expanded diseases?
Yes, symptoms are, that's correct. Symptoms are very different from one person to another. And the difference is due to a different size of the repeats, and not only. So patients that are affected by repeat expanded diseases, they inherited a length of disease. Let's say they inherited, for example, 50 repeats. But during their life, this repeat grows and grows and grows within cells. And we have seen that in organs that are more affected, for example, in Huntington, in some regions of the brains, like the striatum, that's one of the regions that's most affected by the disease, we actually have a bigger expansion compared with other tissues and other organs. So there is this role, and also there is for sure a role of other genes, other factors that, or other modifications that we are not fully aware of.
And Stella, I was wondering, all more increase the expansion, more the symptom worsen it, or it's a direct correlation, or not really?
So it seems to be a direct correlation based on studies in different organs. So we have seen, I'm doing again the example of Huntington, but the same is true for myotonic dystrophy, and studies on ALS are still ongoing because it's so hard to actually look at how big is the size of the repeat. But for Huntington, we have seen that, yes, so like the cells in the striatum have a bigger repeat size, and they do degenerate, they do die earlier than our cells, other cells in the same brain. For example, other cells in the cerebellum, they have a short repeat that's very stable, and actually they don't degenerate that easily. And the same in myotonic dystrophy. Muscles and heart have the bigger repeat size. Why is that? Why it happens that those organs develop a bigger repeat? Because all the tissues, all the cells actually inherit the same size. We are born as an embryo with one repeat size. And why is that is not fully clear, but what we are working on is other factors that might actually play a role in those specific tissues.
It could be also epigenetics factor, or no?
It could be, yes. It could be epigenetic factors. So like epigenetic means modifications, chemical modification on the DNA that don't touch the sequence of the DNA, but they are still modifying the ability of the cells to form RNA and proteins or whatsoever. Yes, they could be also.
Thank you, Stella. Another question is, are the drugs that you talk about already in use, or when you are planning, or when can be used this drug?
Yeah, no, it's not already news. We are working still on the mouse models for disease, and we are hoping to move it forward. Results are actually promising up to here, but we are still at the mouse model stage. We are not preclinical stage yet. So when, I don't know, hopefully sooner than later, if the results are promising and if we confirm safety and tolerability for humans, obviously. So there is some long way to go.
Yeah, as preclinical study are.
Yeah, I know.
Should
That's
be.
the time of science, unfortunately.
Exactly. Another question, if we have a genetic disease in the family, can we avoid our kids being born with the same mutation?
So what's possible to be done by families is genetic diagnosis. So pre-implantation diagnosis before implanting the embryo. That's what can be done to actually like choose the embryo that does not have the mutation.
Mm-hmm.
But currently we don't have gene therapy that can go right and correct the mutation.
Okay, understood. Another question from a person here, it's saying, can you tell me a bit of NF1?
Neurofibromatosis. So it's not a disease I'm fully familiar of with, so I cannot really talk much about it. But for sure we can talk more. Maybe if this person wants to contact me, and we can talk more, or I can direct it to somebody else.
Okay, another question, and so far seems the last, unless there are coming. Can you talk about the mutation that causes Down syndrome?
Yes, so Down syndrome is a bit different from what I've presented you up to now. So in Down syndrome, we actually have one more copy of one chromosome, which is chromosome 21. So instead of having two copies of each gene for the genes that are located within that chromosome 21, we will actually have 3 copies. And that happens because when the germ cells are formed, there is not a full separation between the chromosomes. So we will have some germ cells that will have 2 chromosome 21 stacked together. And so when the egg is fertilized by the sperm, we will end up with an embryo that actually has 3 copies of chromosome 21. That's the genetic background, and it has been correlated, this missegregation has been correlated with the maternal age. But the factors that actually determine it are not clear yet. Still studied.
Okay, so if seems that there are no other questions from the public, unless I'm just losing some things. No, for now, there are no more questions, and I will pass the floor to Corrado Paina. And thank you, Stella, for explaining so clearly and so easily these all complicated diseases that all of us in some way heard about or facing with family or friends. Thank you so much. It was very clear everything, and I hope the drugs that you and your lab are working with, it's going in preclinical phase very, very soon. I really
We'll see. Thank you everybody. Bye.
Thanks to Dr. Lanni and Dr. Emilia Luca for your presentation and for answering important questions from the audience. Now, let me introduce Aurora Policaro Giannace, Manager Corporate Giving from Policaro Foundation. The Policaro Foundation is a non-for-profit organization founded in partnership with sick kids and dedicated to building a brighter and healthier community. Please, Aurora, the floor is yours.
Thank you, Corrado, and good evening. I would like to take a moment to thank the Italian Chamber of Commerce of Ontario for inviting the Policaro Foundation to be part of this special event. The Policaro Foundation was established in 2018 and began with our commitment to Sick Kids Hospital by way of the Policaro Fetal Cardiac Fellowship as a way of investing in the future of our youngest and most vulnerable. The Policaro Foundation provides much needed support to the fellows at Sick Kids Hospital for Canada's primary fetal cardiac diagnostic and treatment center. Since 2018, we have welcomed 3 international fellows to Sick Kids Hospital through the Policaro Fetal Cardiac Fellowship. Dr. Laura Vasquez Garcia from Spain, Dr. Hannah Bourne from Miami, and Dr. Cami Gill from California. As our major fundraiser for sick kids, the foundation hosts an annual Policaro Foundation Track Day. This incredible event brings many people together while raising funds for the Policaro Fetal Cardiac Fellowship. Unfortunately, the event is currently on hold during COVID restrictions. Hopefully, we can all get back to the track as soon as possible. With our initial focus on pediatrics when the foundation began, we have grown to also support other numerous causes. The Policaro Group is committed to supporting health and wellness in our community. The Policaro Group recognizes that even small efforts can contribute to big change, encourage employees, customers, and commitments to work together to build a healthier and brighter future. A few of the events which we participate in over the course of the year are Heart Month, which is in February. For every vehicle sold, the Policaro Group makes a donation to the Policaro Fetal Cardiac Fellowship at Sick Kids Hospital. Breast Cancer Awareness Month, which is in October. We also support the local food banks throughout the year. And at Christmas time, we host the Policaro Annual Christmas Toy Drive in all our dealerships and at select Toys R Us locations. We also donate to the pediatric units at the hospitals in our areas of business, the Oakville Trafalgar Memorial Hospital and the William Osler Health System. If you would like to find out more about the Policaro Foundation, please visit us at policaro.ca/foundation. Again, I would like to take this moment to thank everyone at the Italian Chamber of Commerce for making this event possible, and have a good evening. Thank you.
Thank you to our guests for this presentation and for answering important questions from the audience. Thank you all the attendees connected with us for your interest on this relevant matter. A special thanks to the Consul General of Italy, Eugenio Sgro, and the Sistema Italia for being part of this series of scientific lectures. I take this opportunity to remind you the next appointment on December the 2nd, focusing on deafness and balance disorders. To register to the next class, please check the link in the chat. Thank you for your attention and participation. Allow me before we go to remind you that it's the 60th anniversary of the Italian Chamber of Commerce, Canada, of Ontario, Canada, ICCO Canada. Become a member, be part of the family of the Chamber, be part of the network. Thank you again, and thank you to the ICCO staff for their tireless work. Good evening.